Hello and thank you for your attendance today. My name is Katherine Littlefield and I'm very excited to share our novel findings about post-acute sequelae of COVID and how we are connecting this work to the gut microbiome. First, a quick overview of post-acute sequelae COVID-19, or PASC, as I will refer to it through this presentation. This is an umbrella diagnosis used to cover a wide variety of symptoms that persist longer than 4 weeks after disease onset or hospital discharge. Most common types of symptoms are respiratory, cardiac, neurological, gastrointestinal, or inflammatory in nature. Predictors of PASC include female sex, increased BMI or age, and 5 or more symptoms at onset. Anywhere from 5 to 20% of those with COVID will experience persistent symptoms that can follow any severity of initial disease. With current global case rates, at minimum 10 million people are affected with PASC currently, and overall cases continue to increase. We know the GI tract is susceptible to SARS-CoV-2 infection as viral RNA presence in fecal samples have been documented by a number of groups, as well as visualized in this organoid derived from a COVID patient by Douglas Kwan's group just recently. Results from pilot studies of the gut microbiome in COVID patients have been published, each limited by sample size and cohort representation. Nonetheless, significant differences have emerged. All studies to date show decreased diversity in the gut microbiome, and multiple show increased relative abundance of opportunistic pathogens such as those listed here, combined with a decrease of commensal bacteria. Hospitalized patients in particular show a heterogeneous gut microbiome, and probiotic use in these patients is linked to decreased symptom severity and mortality rates. As gut dysbiosis and GI infection persist during disease course and after clearance, this is proposed as potentially relevant to PASC etiology. We recruited 35 individuals with resolved SARS-CoV-2 infection from the Denver metro area, confirmed by PCR at the time. Subjects were separated into PASC and acute COVID, or AC, cohorts based on symptom duration. In line with published predictors of PASC mentioned earlier, our PASC cohort was significantly older and experienced more severe initial disease than AC controls, based on number of initial symptoms and need for hospitalization. To account for this, we compared results of those hospitalized and not within the PASC cohort throughout the analysis. Another significant difference is the duration from symptom onset to sample collection, which is much higher in those with PASC. In this study, we collected questionnaires on both gastrointestinal and COVID-related symptoms, including questions on severity and duration. We collected stool and peripheral blood, from which we isolated plasma and mononuclear cells. Similar to our group's work in HIV, we are interested in connecting immune function with the gut microbiome and started with SARS-CoV-2-specific T cell enumeration and phenotyping. PBMC were stimulated with SARS-CoV-2 peptide pools of the spike, nucleocapsid, or membrane surface protein separately. Cytokine productions of either IF and gamma, IL-2, or TNF-alpha were used to identify virus-specific T cells and markers of maturation. Exhaustion and proliferative history were included. On the right is an example of flow cytometry data from 1 PASC patient showing frequency of IF and gamma and TNF-alpha production of CD4 and CD8 positive T cells after stimulation. In many PASC subjects, we observed frequencies of SARS-CoV-2 specific T cells of 10% and higher in response to single protein stimulation. First, we looked at the combined responses to all surface proteins and found significantly higher frequencies of SARS-CoV-2 specific CD4 and CD8 T cells producing both IF and gamma and TNF-alpha in PASC as compared to AC controls. As a reminder, the majority of AC samples were collected within 1 month of symptom onset, while the majority of PASC samples were collected at least 6 months from the same time point. Studies in acute COVID show steadily decreasing frequencies of SARS-CoV-2 specific T cells over time. IF and gamma responses were mainly in response to the spike protein. While the frequencies of IL-2 producing T cells follow similar trends, they were not significant. Most strikingly, frequencies of SARS-CoV-2 specific TNF-alpha producing CD4 and CD8 positive T cells in PASC compared to AC were 9.5 and 100-fold higher respectively, while these responses were significantly elevated in response to all 3 surface proteins. Diving deeper into the phenotypes of these SARS-CoV-2 specific T cells, we found higher proliferative history of TNF-alpha producing T cells within PASC subjects as measured by the expression of Ki67. This is data for S stimulation, and N and M responses are also significant following the same patterns. This indicates these cells are actively replicating months after initial infection, and after SARS-CoV-2 RNA is no longer detectable in the nasopharyngeal tract. Like I said before, we also looked at exhaustion as measured by PD1 frequency and maturation by expression of CD45RA and CD27 and did not find significant differences. In light of the contributions of T cells to other inflammatory conditions, such as rheumatoid arthritis, we examined plasma IL-6 and CRP via ELISA. Both markers are also associated with severe acute COVID and with mortality. We found significantly higher levels of both inflammatory markers as compared to AC and positive correlations between TNF-alpha producing T cells and IL-6 plasma levels in PASC. The same correlation was not seen in those with acute COVID, and there were no significant differences when comparing these markers in those with PASC who were hospitalized to those who were not. Lastly, we pulled pulmonary function data that was available for a subset of PASC patients and found significant negative correlations between forced eviction volume, both total volume and percent predicted, with the frequency of IF and gamma producing SARS-CoV-2 specific T cells. This same trend was observed in TNF-alpha producing SARS-CoV-2 specific T cells. There was also a positive correlation between the frequency of S-specific IF and gamma producing SARS-CoV-2 specific CD4 and CD8 positive T cells with the duration of respiratory symptoms that subjects reported. In this study, we observe higher frequencies of SARS-CoV-2 specific T cells in PASC that persist months from symptom onset. We also observe elevated systemic levels of inflammatory markers that associate with T cell frequencies. SARS-CoV-2 specific T cells also strongly associate with lung function. We hypothesize these T cells are associated with viral reservoirs, maintaining these prolonged responses and contributing to disease etiology. With the stool samples we collected, we're interested in investigating the role of the microbiome in PASC and if gut microbiome composition affects susceptibility to viral infection in the gut. I would like to extend my gratitude to everyone at the University of Colorado Anschutz and UC Health who have contributed to this project, as well as to yourselves for listening to this presentation. Thank you so much.